Assalamu alaikum wa rahmatullahi wa barakatuh. Dear students, welcome to our class. So, inshallah, we will today continue from this topic. Last class we stopped till here. So, in this topic, we will see uh, about the contribution of Johann Mendel. He is called as the father of genetics or founder of genetics. So, he is from Austria. What he did was that uh, he in his lab or in his garden he took pea plants. If you don't know what is pea plants, Google, you will know what is pea plant. So he grew them for many years and he compiled a lot of data. Okay, he gathered a lot of information and he analyzed it and he postulated, he made certain laws of inheritance. Inheritance is basically a plant is there and it is growing and then it has seed and the next generation is coming, the next generation is coming. So basically how certain characteristics, for example, seed color, flower color, or these certain characteristics, how they are inherited to the next generation. So that is what is called as loss of inheritance. How certain characteristics are inherited to the next generation. So his work with the pea plant was very important, which laid the foundation for the genetics. So now we will see his laws of inheritance. His laws state that every feature or character, for example, if you take a plant is there and has a flower, okay, and the same species one of the plant will have some uh, yellow color flower and some plant will have white color flower. So this is due to the factors are present in the that plant. So every feature or character, for example, color of flowers, height of plant or color of the plant or texture of the seed, how the seed looks and texture of the pods, the outer surface of the seeds or the location of the flower on the plant. So these are the feature or characteristic of the plant. So he stated that his law stated that it is controlled by pair of factors. So two factors decide this character of feature. So during the formation of gametes, you have the male gamete and the female gamete. So one factor goes to one gamete and it's paired to another gamete. Okay. So so he says like two factors there. Okay. So let's say this is male and this is female, and so during the formation of a new gamete, okay, new generation, new gamete, so one factor from this goes here and one factor from this goes here, any one, okay. So so thus the two factors of a pair segregate or separate during the gamete formation. So these are pairs, okay. But during the gamete formation, new gamete is forming for the next generation. So they separate. So one is separated and one is separated from here. So then upon fertilization, the combination of factors. So these two factors, those combined, right? So this combination, this new combination expresses the future. So this is the first law, his first law. And the second law is that out of the two factors controlling a certain feature. So there are two factors in the new gamete you have. For example, I'll draw here. Two factors are there, okay? Factors is basically, we will see later, we will refer this as gene, okay, gene. Now you understand this as a factor, okay. So out of the two factors controlling a certain feature, for example, two factors there, the dominant one may express in spite of the presence of other, okay. So this will be one factor, another one will be this factor. So let's say that this factor determine decides that the flower will be yellow color, but this factor will decide that the flower will be white in color. If any one of it is dominant, okay, you know, dominant and recessive. If you don't understand this word, check the dictionary. So dominant, if yellow is dominant, what will happen? Even though this factor is found, this white factor is found, still the flower will look yellow in color. So if yellow is dominant, then it will be yellow in color. If white is dominant, white, the flower will be white in color. So this is his second law, okay. Second law, let's say that no yellow is there, only white is there. So white is the recessive here. For example, I'm giving the example. So now there is no dominant factor. that is yellow. So now it will look yellow in white in color. Okay, since there is no dominant factor, so this is termed as recessive. Okay, so that is his law. Then there are laws like law of parental equivalence, law of independent assortment. So we are not seeing all those things here. Okay, so you can read about this. So later, what happened is that Mendel called that as factor. Okay, he selects there is one factor which determines this. So later, scientists discovered that it is a gene. Okay, they said that the gene replaced the term factor. So they later started using the term gene. Okay, so it's like two different genes determine this the characteristic or feature of the plant. So next topic is chromosome and genes. So genes are responsible for heredity. Okay, as we told here, the factor here. So that is genes are responsible for heredity. So where are the genes present? They are present on the chromosomes at fixed points. So chromosomes are there. So genes will be there in a certain fixed points, not randomly. They'll be there in the fixed point. So what is chromosome? You have the cell, and every cell it will have nucleus. Okay, and it contains a fixed number of chromosomes. So every organism it will have a fixed number of chromosomes. In in the cells of the eukaryotes, the chromosomes show the following typical characteristics. So the chromosomes will have certain typical characteristics in the cells of the eukaryotes. So what are those characteristics of the chromosomes? So they are present in pairs. So they will be present in pairs, not single but pairs. One from the father, one from the mother. So one will come from the father, and the one from the mother. So they can be seen only during cell division. So without during the non-division dividing cell, they'll be there in this nucleus, but they'll be in a jumbled up network term chromatin. So they'll be as chromatin. Only during the cell division they can be seen separated. So the paired chromosomes are present in a fixed number, as we told. So they are in the fixed number, and this pair is called as diploid. So this pair is called as diploid, and there is a formula called as 2n. Since it's diploid, so they have given a formula called as 2n. Okay. So each chromosome is made up of one molecule of the chemical called DNA. So chromosome is there. What is it made up of? So you can see here, this is the chromosome, and it is made up of DNA, deoxyribonucleic acid, okay, and some proteins. So before cell division, what happens is that the DNA molecule of a chromosome replicates. Replicate means it multiplies. That is, it multiplies. That is, duplicates to give two molecules of DNA. Okay, so which are called as chromatids. So these are the chromatids. So it multiplies before the division. So why it multiplies? Because it has to be uh, it has to go from the original cell to the new cell during the cell division. So these two chromatids of a chromosome remain attached to a point called as centromere. So they are attached to a point called as centromere. Okay. But in bacteria there is only one chromosome. There's no two chromosome, but only one chromosome. So there is only one molecule of DNA. Yeah, this is how it is in the bacteria. Since there is no well-formed nucleus, so that area in the bacterial cell is called as nucleoid. This region is called as nucleoid, and this is the chromosome in the bacterial cell. So next is human chromosomes. So every cell of a human being, in the human being, if you take the cells, every cell they contain 46 chromosomes. How many? 46 chromosomes. They can be expressed as 2n. Okay, 2n is equal to 46 as we told. So n is equal to 46 by 2, that is 23. So humans have 23 pairs of chromosomes, 23 pairs. Okay, but total 46 single chromosomes. So if you say how many pairs humans have, you have to say 23. But in general, if they ask like how many chromosomes humans have, you should say 46 chromosomes. Okay. So each gamete will have, see, you can see here 23 pairs. Okay. 23 pairs each. Male and female will have 23 chromosome pairs. Okay, so these 22 chromosomes are called as uh, autosomes. Okay, these 22 are called as autosomes. Uh, 22 pairs are called as autosomes. This one pair is called as allosomes or sex chromosomes. These determine the sex or gender of the uh, human. Okay, we will see more further. So yeah, so what happens is that father is there, mother is there. Both of them will have 23 pairs. So during the new gamete formation, what happens? In the first pair, one will come, and from here, female, one will come. So they will join and form the first pair for the new gamete. And likewise, uh, for the second pair chromosome, one from here and one from here. So that will be the second pair chromosome for the new gamete. So same like that, a new full pairs will be formed for the new gamete. Okay, so here X is there, Y is there for uh, male, and for female X and X. These are the sex chromosomes or allosomes. This is how they are determined. For example, let's say from here any one will come. Now X comes, and from here from the mother two X chromosomes are there identical. So one X will come obviously. So now if 2X, then the baby will be female. If the baby has to be male, then the pair should be X and Y. So if the father during the formation of the new gamete, if Y comes, okay, then Y comes, from here X comes, then the new baby will be male. So this is how whether the baby will be male or female is determined. Okay. So these homologous chromosomes, this the same identical pair is what is called as homologous chromosomes. So as I told, 22 pairs are called as autosomes, and the 23rd pair, okay, XX in females and XY in males are called as sex chromosomes. Okay, then the next topic is genes. So genes are present on chromosomes, as we told. So according to Mendel, he called as factors. So they are the Mendelian factors. They are present in pairs, as we told. They are present in pairs. One received from the father, another from the mother on the chromosomes. Um, that you have to understand. So this is the appearance of chromosome and genes. So you have the chromosome, and this each arm is called chromatid, and here you have the centromere that connects this chromatid. And if you unwind this, then inside this you will see the DNA. DNA is a double helix. So you see one and another one here. See, this is double helix. DNA double helix, and inside it will have bases, sugar, phosphate, and this is the gene containing nucleotides. We will see about this in the upcoming topics. So genes are present on chromosomes that are called as segments of DNA. Okay. So next is, so basically genes, they carry the information that will be passed to the next generation. Okay, they will carry the information that will be passed to the next generation, which contains the factors, that is the characteristics. For example, if fathers or mothers, anyone's eye is blue in color or greenish eyes or brown eyes, okay, then this is due to a gene responsible for that in their body. So if the son also has brown eyes, which means that which is inherited from either one of them. If the father has brown eyes and the mother has black eyes, if the son has brown eyes, then we can be sure that he inherited this from his father. So, in our body, each and every characteristic, whether it is of hair, how he looks, for everything there are genes. Okay, so now you can get what is genes. So genes, they store this information. Okay, So there is something called as DNA fingerprinting. You can read about this. So in the police industry, to find out the criminals, what they do is that they do some DNA test, and which is called as DNA fingerprinting. So just like in the past, they used to have some fingerprinting method. Okay, they had a some method through that they find out the criminals. So in the uh, modern police this thing, to find out the criminal, they have this modern feature called as modern method called as DNA fingerprinting. So each DNA, each individual have unique DNA. So that you have to remember, each individual will have unique DNA. So in the crime scene, if the criminal leaves of hair or drop of blood or semen of the criminal is left, and they will go to investigate, and if they find out, they will do a DNA test and they will compare with the suspect. Okay, suspect, you know what is suspect? Like they'll have like suspects of criminals, right? They will do the genetic test. They will take out the gene from them and they will do the test and compare. If it matches, then they can be sure that that is the person who did that this thing act of criminal act. Okay. The next one is DNA molecule. So what is DNA molecule? As we told, it's a polynucleotide. It is made up of poly means many nucleotide. It contains nitrogenous base, a deoxyribose sugar, and a phosphate. So this is what DNA has inside. And when it comes to the nitrogenous bases, it has four nitrogenous bases. One is adenine and guanine and thymine and cytosine. You have to remember this. AGTC. AGTC. Adenine, guanine, thymine and cytosine. These are the four kinds of nucleotides in a DNA molecule. Okay, so they will have different combinations of these nucleotides in a segment of DNA from the different genes. Okay, so next one is DNA replication. The cell division takes place in a manner so that one cell divides into two identical cells. Okay, cell is there and it divides into two identical cells. Okay, and it will have same number of identical chromosomes. So this, for example, let's say that it has a chromosome. It has, for example, humans, it has 23 pairs of chromosomes. So it has to divide into two new cells. What will happen? This will produce one more copy, okay, and it will be divided equally. Okay, that is what is mentioned here. So this is either called as DNA duplication or DNA replication. Before the division, the DNA has to be duplicated so that it can be divided equally into the different cells. Okay, so the double-stranded DNA molecule unwinds. So it is DNA strand. You can see here. Okay, it has to unwind. Okay, with the help of enzymes to expose the two strands of DNA. So two strands here are exposed, and with the help of an enzyme called as DNA polymerase enzyme, which forms a new daughter strand. So you can see new daughter strand is formed here, the golden color here, with the help of a polymerase enzyme. So finally, new two DNA will be formed. So this is how the DNA replication is formed. This is the structure of DNA, double helix. You can see two helix are there. Okay, and inside that you can see the different nucleotides in a different combination. So ATGC is there, right? A will bind to T and G will bind to C. So adenine will bind to thymine, guanine will bind to cytosine. So, but it will be in different combination. AT, GC, GC, AT, like that. Okay, in different genes. So we'll stop till here. Inshallah, from the next class we will continue from blood group inheritance in humans. Okay. So study well. We have to complete the portions as soon as possible. That's why I'm teaching fast. So if you have any doubts, you can ask me. Okay. See you in the next class. Assalamu alaikum wa rahmatullahi wa barakatuh.